Hello, everybody. This is Peter Cooper from the NCBI. Welcome to today's webinar. Today's webinar is about how to find data for your research organism, meaning your non-model organism. Adelaide Rhodes is going to be our presenter today. So let's go ahead and get started, and I'm going to turn it over to Adelaide now to start presenting. Take it away, Adelaide.
Okay, thank you, Peter. So I'm a recent addition to NCBI, and I come from a background of studying marine crustaceans that are found in the benthic zone in the ocean, so down in the mud. And there isn't a lot of genomic information about them, so I started wondering, how can I get the best information on a research organism that isn't a model organism? And I thought that might be useful information to talk about today. So what we're going to cover today is just a basic introduction to the entry points to searching at NCBI. There's more than one place you can go to find data. And then the second thing we're going to do is a demonstration on a well-annotated organism, a whale, whales, and looking at a specific gene. And then we're going to run through a few exercises such as finding sequences, making a set of primers, and then finding other related sequences that may not have shown up in the direct search. And then we're going to do a second demo about unusual organisms to show where you can start at NCBI Taxonomy Hub in order to browse data sets and get meaningful information about your genes of interest. All right, so it's good to establish what types of records can be found at NCBI. Most of what we have is submitted. So the audience, you guys, are submitting your sequences as you go through your research programs, and it's there's a lot of sequence in the database now, several petabytes of data of sequence submissions that are either coming in through GenBank, the European Nucleotide Archive, or the DNA Databank of Japan. So they share the resources of what is being submitted by the researchers. And then also we have something called the Sequence Read Archive that holds all the raw next-gen sequencing data. So there is a difference between stuff that is submitted and stuff that is curated. So you may have heard of RefSeq. Really what that means is NCBI itself, the researchers and educators behind the scenes have taken the records that have been submitted and curated quite a few of them and then put them back out into the record database with a link back to the original submission so that you can have confidence in what you're looking at as being a good definition of a gene or a protein boundary and things like that. So those tend to be a little higher quality or more reliable. But there's a lot more to it than that. We have literature databases such as PubMed. There's other types of molecular databases, like, for example, variants, dbGaP. There's population sets or geodata sets as well. And we have webinars on all of these topics. So please, feel free to look through our YouTube channel to find more detail on all the other databases. But the two places I'm going to concentrate on today within our Entree system, which is what is called like our large collection of databases, are two central hubs, gene and taxonomy. So I'm going to show you what gene and taxonomy look like when you need to get information on a research organism. So this is the front page of gene, and I could have gotten there, through Entree, or there's a URL here at the top of the page. So you can come into gene, and I like going to the front page to point out to people there's a lot of helpful information, such as guides on how to use gene. So there's a gene quick start over there on the left. There's also download options. You can get on a mailing list. There's all kinds of things you can do from the front page. But it also defines what kinds of records you're going to find here. So that's the first data hub. The second data hub we're going to spend a little time in our demos called Taxonomy. And as you can see, it also has a quick start guide, and it has different sets of data that it provides. It's organized by the classification of all the organisms in the public sequence database. So a natural place to start if you're not working on human or mouse. Right, and then the third thing, this is a slightly newer interactive browser set. It's called Datasets. It's in beta testing, as you can see, the little yellow sticker up here, beta testing. Don't know if you can see my cursor, but anyway, it's a rapidly growing database. Right now, it's limited mainly to eukaryotes and reference sequence genomes, but also SARS. COVID is a big deal, so they've added that. And there's always announcements happening about what's available now in Datasets. So you may see a Twitter link, like the one I've taken a screenshot of here, where it says there's more annotations now. So it's a good place to go if you're looking for annotated gene data sets for organisms that are, not your typical model organisms. And as you can see, last year in May, there was one for that included some killer whales, bumblebees, starfish, etc. So it's a great little feed to follow in order to see what's new. Okay. And then, of course, if your gene is not found, but like if there isn't like an annotated gene set, there's ways to get there using a couple tools. So we do have BLAST. I think most people who think of NCBI think of BLAST first. It allows you to compare sequences across the entire database. So that's quite a handy tool. And then, of course, we also have primer BLAST now. So if you have an organism that you're researching and you're collecting samples, you would probably want to take advantage of the ability to design primers from very similar organisms that have genes of interest. So we're going to walk through that as well. So I'm about to transition to the two demonstrations. The first one lasts about 10 to 15 minutes, and the second one is a little bit shorter, but be happy to stick around and answer questions after. The first demo is looking for a gene of interest, myoglobin, in whales. And what we're going to do is get a set of sequences from the Gene Data Hub that I showed you. We're going to download those sequences from Datasets, and then we're going to use the sequences we downloaded to make a set of primers. And then we're going to go back to the original gene search and try to expand it by doing a blast on one specific whale. And then in demo 2, we're going to start in the Taxonomy Data Hub. I'm just going to show you guys a little bit that you can also get jellyfish sequences from Datasets. We're going to do some blasting there, but in a different way, slightly different way, just to show you that that's available. And then we can show you how you can annotate a gene of interest. All right, what is myoglobin? So I've said it a few times in this presentation. It's a oxygen storing protein in mammals' muscles. So when whales and seals and dolphins need to hold their breath, not all the oxygen is carried in their lungs. They can pack a large amount of oxygen into their muscles, which increases the amount of time they can spend underwater. And this is an area of scientific fascination. There's lots of papers about it. I have just a little note over here about a publication on a beaked whale that set a 4-hour long dive record, which is the longest known incidence of a whale holding its breath underwater. And it was a beaked whale. So that's a particular type of whale. Has to do with the shape of its face. So so that was really interesting. So this is an area of active research. So there is data available to find. And then the demo 2 that I'm going to work on is looking for jellyfish. So what if, I'm not working with what we like to call as invertebrate people, the charismatic megafauna, the big guys. Where do I go to find my data? So there's going to be a second approach looking at the available data using NCBI taxonomy. And then we're going to look for some conserved genes in an assembly that is not annotated. And the organism I chose for this demo is called Sandaria meliensis. It's a very cool looking true jellyfish. I mean, it's a scyphozoan. And so I'm going to now go into the demos and show you guys what's going on. All right, so demo 1, I land on the gene page and I want to know what informations they have for whale and my gene of interest, myoglobin. And when I pull it up, I'm going to see that I got 15 results, okay? But the first thing you might want to do is make sure that your search, as you can see here, I had to capitalize, and that's a quirky thing about NCBI. You want to make sure your search gives you what you're looking for, and it's not accidentally stumbling upon something that may be named whale, but not actually whale. So if you scroll down here, you'll see that, yes, these first few ones are definitely what we would consider whale, to be whale or dolphin. But when I get down here, I see whale shark. So whale shark's not a true whale, it's a fish. And also, I got a bacteria. And it just means that whale was too broad of a term because it looked for whale over here. If you look in the search box over here to the right where I'm highlighting, it looked for whale in all the fields. So basically, each record has a title and a description, and it has the authors who provided the record. And if one of those fields also contains the word whale, it may accidentally show up in this field. So there's a few different ways to correct this. There's a taxonomic grouping up here, so I could simply just click on mammals to get rid of the fish and the bacteria, and that would give me my whale list. Or there's another way to correct this. I could, so that was up here, if you guys can't see my cursor. Or in the search detail box, I could get rid of whale in all fields right there and then resubmit the search. Or what you need to know, and one thing I was reminded of as I was prepping this presentation is if you just put the plural version of what you're looking for, so whales instead of whale, and I rerun the search, it actually gives me just whales. And you'll see that the gene page, the results page, has changed slightly. So now you see the taxonomic, name is Cetacea, which holds the whale groups. And so you'll see over here that the search box has been altered to be correct. And now my taxonomic grouping is only whales and dolphins. So that's just something to be aware of if you're not really familiar with gene or how to search for organisms. And then I just want to quickly say up here, if you get really lost, you can choose to add, qualifying things here like organism. This is another place I could have put whales. That was in the advanced search dropdown, which is up here. Okay. And it's, as you saw, like when I went over there, it saved all my previous searches, right? Okay. So now I have a good data set. What can I do with it? Like, where am I going to head with this data set to actually get these sequences? And so this is where I'm going to highlight our new interactive website that allows you to download easily called Datasets. But in order to do that, I need to get this table. This would be really hard to just kind of copy paste over. It has like special figures and checkboxes, but luckily, if you look down here at the bottom and click on tabular, you can just get a list of the unique identifiers. So these are the records that are identified in that table. And so I'm just copying those records. So you can get a table, you get a summary, you can get all kinds of things, but the easiest way to go from here into the next section of NCBI I'm going to show you is by copying that. And now I'm going to go over to Datasets and use that information. So here is Datasets. So I think it's a very beautiful website. I love seeing the pictures of the organisms I got up there. And just to quickly show you, you can get genomes from going to Datasets. You don't have to go through the regular NCBI interface. And, it's a great way to kind of pull up whales here if you want the whole genome. So that's a lot of records. Like I started out this, demonstration. I only want one gene, from whale. So here they've actually got it broken down into more, things, but it's Cetacea. So I could find out what genomes are new and interesting. So we've got some beluga whales, we've got gray whales, things like that. And then there's some genomes that are categorized as reference, meaning they're a little bit more annotated than what's been submitted. But this is cool to know about if you want the whole entire genome. But we came over to Datasets actually to use those unique identifiers. So I'm going to go back to the front page on that. Come on. Here we go. And I'm just going to scroll down a little bit. So here's the front page. I'm scrolling down. It's right here, Get Started. So under Get Started, I'm going to enter my identifiers manually. I could have had a file, but there's only 12. It was easy to copy paste them. And so when I open it up, you'll see, oh, the information I got from the part of the database called gene, that's the first option. So I'm going to copy and paste that. But there's other options here. I could do gene symbol, accession number, but I had copied that from that table. So I'm going to add that. And boom, there we go. There's the sequence information that I can download as a table or a set of FASTA files. And what's cool about this, for those of you who've spent a lot of time on NCBI, is I don't have to go to the protein page, get the proteins, and then I don't have to go over to the nucleotide page to get the nucleotides. They're all here. And you can get to them, you can see them by switching transcript view here. But another cool thing you can do is add more information. If you want the table, like if you're not actually going to get the sequences, you just want the table, you can get the table to be a little more complete. You can add common name, for example, right? So killer whale is orca over here. Another thing to notice is we're in gene view, and it shows that there's 2 entries for orca. So there's more than one gene view. So I'm not sure if that means there's paralogs or stuff like that, but that's kind of cool too. And you'll see there's 2 different links here. So there's 1099 and 1162. So I may only just want one sequence for each organism. So maybe I'll switch over to transcript view, which instead of having it, here you had to click here to open it. But when I switch to transcript view, it gives me both versions, right, of every transcript that leads to the gene of interest. And so maybe I only want the longest one. So that's one of the columns that I've selected to open. So I select all by clicking on this checkbox here, but now I'm going to go back and unselect anything that is the shortest version of the gene. So here monodon has 2 versions. There's 671 and 744. I'm unchecking 671. Globicephala, also unchecking 1061. And then, there's one more here. Okay, great. And so now I'm ready to download. So this is where I can go here, download, and I could just get the table, but I really want the data set. I really want the FASTA, and I just really like this a lot because it pulls up genes, transcripts, and proteins all at one place. And so I can call it whales myoglobin. Okay, and longest. Okay. And then it's going to download. And here I'm just going to make a little note because it is in beta. It's going to, if you're in Chrome, it will actually download a zip file. Don't be surprised if you're in Safari or Firefox. It may not work. So just switch browsers if it doesn't actually download a file. And then I'm having a little issue with unzipping, so I already had unzipped it on my computer so I could show you guys what kind of information you get. So sorry, I'm just going to switch over to here real quick. So this was the zipped file. I unzipped it, and when I open it, you can see it may change the name of your folder to NCBI Datasets. So that's something to be aware of. So if you're doing this a lot, make sure to rename your folders to keep old datasets. And then under here, it's going to have my protein FASTA and my RNA FASTA here. And so the RNA FASTA is what I'm going to use next in the demonstration, because I'm going to take this information I downloaded from Datasets, and I'm going to go over and get some primers going. So here's the FASTA file. And I'm just going to, it's small enough that I don't have to use it as a file. I'm just going to copy and paste for this demo, but you could do files. And now I'm going to go to the next website I'm going to demonstrate, which is called Primer Blast. And all of these links can be found in the slides. But also it's really easy to Google for these things. You can just Google Primer Blast NCBI. It'll bring you right here to this website. So now there's 2 things you need to do. You need to switch this tab here. So this is a 1 template. So if I only had 1 sequence to enter into the box, I would use this tab, but I really need to switch it because I've got multiple sequences. And then I'm going to click on this, right? And it's going to load, and from copy paste, or like I said, I could have chosen a file. That's one, is switching the tab. And then two is I'm working on whales. I don't really want to see human primer sets. So I would come down here maybe and change this organism. And you'll see, like, if you start cruising around NCBI a lot, this box to choose your organism is everywhere. So like, take advantage of that when you're working on a non-model organism. Set the search parameters to be just your organism of interest. Don't let it search the entire databases, because then you'll be frustrated by a lot of searches that go off to human or mice. Here's another place, like, you can make changes. Like, there are drop-down boxes at the bottom. But we're ready to go. So I'm going to click Get Primers. And it takes a couple minutes to run. Come on, is it running? Why isn't it running? I'm afraid this would happen, but that's okay because I already saved the results elsewhere. It works. Believe me, it works. It's just, of course, it's not going to work while I'm sitting here, but I did save the results. So basically, what you're going to see when you design a primers against a bunch of templates, it's going to give you more than one option. But what it did is it took the accessions that I entered, those FASTA files, it stacked them up all against each other, and then it designed the primers up here. And so you can pick a primer set that increases the probability of observing the portion of the gene that's of interest. So if you look at a lot of these, sequences compared to each other, you'll notice that they are different just in certain areas. So you can kind of see these red spots here. So maybe you only care about protein changes that happen in this particular region. So you'd want to put like maybe primer set 9, or maybe you care about gene changes over here in this particular region. You'd pick one of these primer sets, right? And then down below, things like melt and statistics and stuff like that have a lot to do with how you run the PCR and your thermocycler. So all the details are down here so that you can get the best result when you do actually use one of these primers as a template. Okay. All right. So that's Primer Blast, which is kind of a special way to use data. But now I'm going to go back and point out something about the original data set that we were looking at. So I'm going to have to reverse back. Okay, so when we were looking at data sets, I might want to look for other sequences that are not reference sequences, that are like a little less annotated, that may have just been submitted recently, or haven't been identified as myoglobin necessarily, or just are predicted. Something to notice here is that there's one sequence here that's different than the others in this list. And what it is the sperm whale has an accession number that begins with the extra special N. When you see an N, that's actually really good news. That means there's been some extra work done to validate that this is the gene that we think it is. And so an X is a predicted protein, even though there may have been some work to validate it with some RNA-seq and things like that. It's more in silico prediction versus like a study that actually validated. So I just want to open this up to show the difference here. So somebody was, using, there's several different journals that, talk about this particular sequence that are showing how they validated it. So, they did some crystallography, crystal structure, they've done other things to prove that this is the protein that we think it is. So this is a great sequence to choose if you're going to do some more blasting for related sequences that aren't necessarily in the RefSeq database. So I went over to the record here. I can run BLAST directly from here. I could just grab the accession number, to go to BLAST. There's many different ways to do it. So if I go to the front page of BLAST, it'll give me a few choices. So it is a nucleotide sequence. So I'm probably going to do a nucleotide to nucleotide thing. Later on, we're going to do a protein to nucleotide search. So that's going to be T-blastin. But today, right now, at this time, we're doing nucleotide. So I can just enter that accession number. So it's the sperm whale version of myoglobin. Okay, so as you can see, I've been running BLAST and practicing for this, so it recognize that my organism, but it would say human probably here for you guys. So what you would want to do is to change it to whales. You can just start typing in whales. You could put in cetacea, right? The taxonomic ID won't change. So it's always 9721, whether I put in whales or cetaceans. It's really, this is like the number that BLAST is using to kind of limit the amount of search. And then down here, I want to kind of look for things that may not be annotated very well. So I'm going to just say somewhat similar BLAST. And so that's like a little bit, looser. And then another thing I like to show people who work on non-model stuff is that you don't have to just go with 100 hits. You could increase the amount of hits you get in your BLAST search. Like I could up it to 250 if I wanted, because the more distantly related organisms that have just changed enough may not get a high hit value, but they may be in there. So by kind of increasing the number of hits you get, that it maximizes your chance of finding interesting stuff. So I've set it up. I'm going to click BLAST, so please work this time. Okay, good. So this, my BLAST is running, and it takes a couple minutes, not terrible, so we'll just let it run so you guys can breathe. Okay, great, boom. So here is my list. And so it keeps my recent searches, for a few days, right? It keeps it until the 12th. So that's not too bad. So like if you want to come back like the next day and keep working from a place that you left off of, you can use this little link here, recent results, and come back to these results. But as I scroll down, you're going to see now this list of organisms that we can see, and I can pull up common names again so we can see, are a little different than the list that we started with. It has added species by doing a regular BLAST, and you can see here that some of these are just submitted sequences coming in from all over the world. Once again, like Europe and Japan and US are the main three, accession number types that you'll see. And it's in our database. So now we have a much longer list of interesting sequences, but there's a few things instead of just plain old downloading, right? Like I could just grab the sequences. I could select all the sequences and then I could just download them here using download, there's things you can do interactively. So I want to look at maybe how closely related some of these things are. So I could either use taxonomy, right? So taxonomy, the tab up here, opens up the list of things that got a hit. And then I could go to just like, I can be like, oh, I really care about this particular species. I can go just directly to that hit by navigating through the taxonomy tab. Let's see. So if I go back to descriptions, I also see there's options here to generate some distance trees. So like if I use distance tree up here, it grabs everything, but maybe I only want things that have really high coverage, right? So I want to make sure that it's a decent chance of it being the thing that I think it is. And I also want it to be kind of long. So I just arbitrarily said, I'm going to get rid of, I'm going to uncheck the species that have slightly lower coverage and let's see, and are slightly shorter here. And then I'm going to generate a distance tree of results. And so this isn't like a true phylogenetic, analysis tool, but what it does is it kind of just organizes your findings. You can just get like a quick glance, like what's going on. So here you can see that things are like this is the pygmy sperm whale, and then here's sperm whale. So those are kind of outliers. And then Peter Cooper, who's also on the call, showed me this cool way to, make it look a little more interesting. If you go over here to tools, there's different layouts you can choose, such as a radial tree that kind of boom, like spreads out the species names. So it's a little easier to read, like who's with who, right? And so, we have the Balaenoptera down here, and we have the pygmy sperm whale kind of closer, branching off of sperm whale, right? And then something very interesting, if you recall, like forever ago when I brought up about that article, these are beaked whales. So the beaked whale that was in that article was called Ziphius, I guess there's not a sequence that showed up in GenBank for that yet. You know, it was a more of a nature study. It wasn't really a molecular study, so the sequence may not be available yet, or maybe it's private and not submitted. But you can see the other beaked whales kind of branch off by themselves as well. So sperm whales have a kind of a slightly different myoglobin, and apparently maybe beaked whales do too. So this is just kind of a cool way to confirm, you know, your viewpoint, like before you keep going, and you know, is this interesting? And so yes, you definitely would probably want to download these sequences and maybe do some, you know, there's some duplicates once again, you know, maybe you would screen for different things. And then we're not really going to talk about it today, but you can do alignments. Like here, this is the alignments against the query sequence, but alignments here, that's something you may be familiar with. It's more like a muscle or a clustable W, but it's just a quick and dirty version. And like, we're not going to spend a lot of time here, but I just want to show you that that's an option. It can line up and show you where your sequences are different from each other. Okay. All right, so that was demo number 1. Let me just bring back up the PowerPoint. So that was demo number 1. We talked about the beaked whale. Now I'm going to move into demo number 2, which is a little shorter of a demo. Okay, great. So just to remind you, this is going to be an organism that doesn't have a lot of annotated sets in the NCBI, except for a few particular species. Like if you're working on corals, there's a lot of annotation available. So that's a good place to start if you're looking for genes. But if you want to work on jellyfish, there may not be as many locations to get that information. So let's go the other route of getting information for my research organism. So here I am at the starting point called taxonomy. And you can just get here by Googling NCBI taxonomy. Sorry, my dog's barking. And so here we are in taxonomy. And I might want to start by going, well, I want to work on jellyfish. So what I've learned is it's better to put the plural version of a name, a common name in. And it's interesting, it actually brings us to the Scyphozoa. So it brings me to Scyphozoa, which is the class of true jellyfishes, because there are other things called jellyfish that aren't true jellyfishes. And if I click on that distinction, it's actually going to show me all the families and genus and species that are entered into GenBank, right? So there's a lot of information on jellyfishes in Scyphozoa, right? Like if I wanted to go see the cubozoans, I might want to go back up to Cnidaria, right? So cubozoa are where I believe our Portuguese man of war and stinging things are. But, you know, you can jump around using this classification system that's at the top here, but I want to go back to Scyphozoa. Okay, great. So I'm here, and I want to find out how much information is there about jellyfish. So you may notice this little grayed out box up here. It has all the different kinds of data that are carried inside NCBI, but the most interesting data sets to start with usually are nucleotide, gene, protein. You might want to know about raw data sequencing experiments, and then of course genome assemblies, even if they're rough. Like it's good to see a genome assembly, see if something new has been entered. So you can click these boxes and then just go here, go. And when you click on that, you'll see these numbers are showing up to the right of the classification. So for all jellyfish, there are 12 assemblies available. There's 143 genes. That's not a lot, but there's a lot of nucleotides and proteins and SRA experiments. So that's where you go for raw data that may not have been assembled well, but you can still find data in there, especially now that we're getting long read sequences, right? So that kind of reduces some of the confusion of finding bits and pieces of things. So I thought an interesting case study would be a jellyfish here, Sandaria, that does have an assembly. So there is one assembly, and then there's 22 SRA data sets. But as you can see, there's not been a lot of annotation done because nucleotide and protein are very low numbers, right, for what you'd expect for something with a genome assembly. So if I click on that number 1, so I just clicked on the number 1 next to Sandaria, it opens up that assembly, right? And it gives me a lot of options I can't get directly from the BLAST page. So I can run a BLAST here. I can run a primer BLAST that's specific just to this genome assembly. So we're going to do that right now. We're going to blast the assembly. And let me just show you how this looks slightly different. So the BLAST page here doesn't have as many options. You don't get a protein blast, right? Because we're talking about genomic stuff. I'm just going to close the warning. It automatically picks the database for you. So there's no organism like box or anything like that. You're just going to be blasting directly against the assembly. Okay, so it limits the size of what you have to do. So now I'm just going to go over here and get the example gene. So this is a housekeeping gene called CDC20, and this is also in gene. Like when I plugged in this name here, let me just show you how I got here. I thought it would be an interesting example. So like here, if I go to CDC20, it doesn't show me all that to begin with, but if I, click on one of the links, it gives me this full record. So a full record for a gene tells you a lot about it. It gives you literature, it gives you RNA by tissue. And so this is generally something you'll find for human stuff, but it doesn't mean it wouldn't help in your search for meaningful genes or which tissues to look in, right? Because if something is in the brain, it might be in the brain or what you're looking at. And it maps out what your expected exons and introns should look like for this particular gene if you are successful in locating it, right? So there's a lot of information about CDC20, which is a part of, it's a regulatory protein that's used at several points in the cell cycle. It requires 2 microtubule dependent processes, et cetera, et cetera. It's a very good check to make sure perhaps, like, is this a good genome assembly, right? It's a eukaryote that goes through this process, the cell cycle process. This gene should be in there. Great. So I just grabbed the accession number. Actually, I'm more likely to get hit if I use the protein, and then I do a translated blast search. So I'm just going to go over here and copy the protein. Okay. So when you're doing a search, nucleotide to nucleotide is a 1-to-1, right? But protein can be translated 6 different ways. So you get, 6 times the options of finding it in a genome, especially one that hasn't been annotated yet. Great. So that's why I'm using tBLASTn. It's a translated blast search for a protein. And now it's going to search the assembly to see if this protein is somewhere in the genome assembly. And the BLAST results here will look different because it's not going to have annotation except for the BLAST result. So the quickest way to get to the answer of whether this is in here is like to look at the alignments page. So you should have seen these types of things before. So it is showing, we did find a lot of homology between that gene, right? And it's in a lot of hits within the same contig here. So when you see a range like this is like where in the contig part of what we blasted against landed. But way to organize the information so you get to the right thing right away is to go here to this box and sort by query start position, which then brings up the query that starts closest to the beginning. Because if you click on the graphics here, it's going to show you this really long contig with the teeny tiny little hits of things. But this will help me organize and pick one that starts close to where my query started, right? My query position 6. But it starts at position 1.4 million, like inside the contig. So this, click on this GenBank versus this GenBank is going to take me directly there to view it. And so this will just take a second. Oh, I meant to click graphics. I hit the wrong thing. Graphics, I meant to say graphics there the last 2 times. Okay. So I want to look at the graphics, but also the GenBank, brings me to the part that I care about in that contig. Okay, so I zoomed in on just the first part of where the hit is, but by zooming out here, I can kind of see that this protein I used as a model recovered hits that seem to recapitulate the exons that I'd expect to see. They're slightly fuzzy here, and but they're all the exons are there. So this is a great way to view the quality of your assembly and also to get started on finding genes of interest in your organism. And that's it. That's all I was going to talk about. I mean, there's other things. Just really quickly, you can zoom in here, and it gives you a bunch of options of what you can do with just that range, right? So now we've located where it is in the genome, but it's in this really long contig. So we could go to the range. We could blast search on just the piece that I've got highlighted in green. I could do a primer blast, or I could just get the FASTA and then maybe use that as a search tool for other jellyfish that had information for them. So I think that's where I'm going to wrap it up. I've been talking a lot. I'd like to hear from the group about their questions. So let's go.
Thanks, Adelaide. Okay, so there's lots of there's some pretty specific questions that I don't know that everybody is interested in the answer. One of them that I thought, Adelaide, you might have some input on because I don't really know. One person asked about annotating, like this, so they have FASTA sequences and they want to annotate them. We do have a prokaryotic annotation pipeline that is portable and you can set up, but I don't really know what people do out in the world who don't have access to a eukaryotic genome annotation pipeline. Do you have any thoughts about what kind of things would be available to people for free? Because he's interested in a free thing.
Yeah, there's a couple open source genome browsers, right, that you can build. I believe UCSC has one.
Where you can annotate? Where you can do annotation.
Yeah, you'd have to know command line for that one, but there's one called JBrowse, I believe, that you can use. I would just do like a Google search for genome browsers, like free genome browsers, because a lot of those are open source tools, and there's even websites that you can go to and load your genome of interest. So UCSC lets you do that. It's complicated, but you can. Like, there's ways to upload your genome of interest or pick one that's already been done. So there is a link over from NCBI, and then you can save your annotations as files there. Yeah. You can annotate for yourself and save the annotations. You don't publish them there, but that would be like where you could, when you publish your, these people here like didn't annotate the genome because they were using it for something different, but many people do end up coming back and publishing, their findings, and that becomes records in NCBI. Yeah. Okay.
Another question that, a comment from one of the people in the questions pod. Someone mentioned an algorithm called Augustus. Are you familiar with that one? That's related to the question.
Oh yeah, yeah. I'm pretty sure that's free. So yeah, that'll give you a file. That will give you some output with some like genome coordinates that give you its best hits. Yeah.
So that's called Augustus, like the emperor.
Yeah.
A couple of other questions that I think I'll address, if you don't mind. One of the things that people asked several times in the chat was about how to find their particular type of sequence. And one of the things that Adelaide pointed out is that if you have a particular organism in mind, use that organism's name followed by the word organism in square brackets, and you will get all the data for that organism. Otherwise, or you can also go to taxonomy to do that. And then whatever particular gene or sequence you're interested in, then add that name to the end of your search, and you'll get what you get when you go into the nucleotide database or into the protein database.
Here's something I do. Like, I'll go to the main Entree gene page, and I'll look at all the databases. And so maybe I care about porcupines, and I'll put organism here, like Peter suggested.
I don't know if it's going to be one or not. We can try it. It might be.
Yeah, why not? yeah, so it didn't work, but maybe what else could I use here?
So you can try searching porcupines in taxonomy and see what it does, because sometimes it's helpful. Like say,
okay,
see if that works. It may. So change that to
yeah, but
taxonomy.
that's one place I look sometimes is using common names, like Peter's pointing out, sometimes it works better.
So let's see if we
Okay,
got anything. So there are a bunch
there's
of there actually is there's a family. There are two different families of rodents that are porcupines. There's New World and Old World, and so you'd have to know which one you were interested in. But if you knew that,
right.
then you could go there and do that kind of thing to get your. I mean, if you click on that, of course, then you'll get that table of all the data.
Oh yeah, that's a good point. We should point out the table over here.
Yeah, and so then if you say you want to get all the nucleotide sequences for this family, then you could simply click over there to get them. And then, if you wanted to add something, so suppose you're interested in a particular gene in here, you can just, write the Boolean operator and at the top,
Mm-hmm.
whatever you want to do, whatever gene might be in here. Who knows? You'd have to look and see. This is a good example of a group of organisms that's largely been probably explored for taxonomy purposes or for, you know, for doing phylogenetics on or something like that. So you're going to see a lot of marker sequences probably.
Okay, cool. Other questions?
Well, there's a bunch in here that I don't think that I have answered somewhat, and we're kind of out of time. So what I think we should probably do is to wrap it up. And I don't know if you had that slide at the end that has the sort of the help document
Oh
stuff,
yes,
but we
thank
could just
you.
throw that
Thank
up
you.
there.
First,
And what I'll
first.
do is we'll answer these questions and make the document available. So just to mention a couple of things here. If you want to keep up with what's going on at NCBI, and we have a blog called NCBI Insights. We also have a number of fact sheets for different resources that are available on our FTP site. So go there and to find out about those, and also our YouTube channel. So if you just Google NCBI YouTube, you'll find the channel. We're moving that stuff over to the NLM YouTube channel, so you might want to check there too. So thanks everybody
Thank
for
you.
coming, and thanks Adelaide for that presentation. That was very nice. So bye
Good.
everybody, and we'll see you next time.